Hey guys, let's do section 8, the cytoskeleton. The cytoskeleton does exactly what it sounds like it does. It is the skeleton of the cell. It gives it its shape. It assists in the movement of the cell and the organelles within it. It makes internal transport possible. It's how things move around within that cell. There are three main types: actin filaments, intermediate filaments, and microtubules. This cytoskeleton can be assembled and disassembled as needed by the cell. It has been compared to the bones and muscles of an animal, and it is dynamic, which means it responds to environmental changes. Let's talk about actin filaments first. They are extremely thin filaments, twisted like a pearl necklace. They make up a dense web that is found just under the plasma membrane to give the cell its shape. They provide support for microvilli in intestinal cells. Those are the cells that absorb nutrients from your digestive tract. So this actin filament gives those places of those microvilli a place to anchor, so that they can stick out of the cell and absorb nutrients from the food as it passes through your digestive tract. These actin filaments are also for intracellular traffic control. They move stuff around within the cell. Think back to the endomembrane system. This actin filament is how those vesicles moved from the ER to the Golgi body to other places within the cell. They are also responsible for cytoplasmic streaming, which is the movement of cytoplasm within the cell. Actin filament also are part of let me talk for a second there, guys. Their function in pseudopods of amoeboid cells, they form what's called a fake foot, which is how the amoebas in pond water move around. And they form an extension of the cytoplasm of the membrane. So it's like it's sticking its foot out, and then the rest of the cell joins it. It kind of looks like walking. Actin filament is also an important component of muscle contraction. So every single time you move a muscle in your body, you can thank actin filament. And these actin filaments are also important in animal cell division, where one cell then splits into two cells, which we're going to talk about later on in the year. Here we have a diagram. Here is our actin filament, these green pearl-looking things. Okay, they are just below the cell membrane. All right, they are attached to the membrane via myosin molecules, and they contract using ATP and cause movement within the cell. Intermediate filaments. They are the middle size between actin filaments and microtubules. They are rope-like in assembly of fibrous polypeptides. You'll remember polypeptides from last chapter. They are long strings or strands of amino acids bonded together. They vary in nature depending on what kind of cell they are in. They function in support of the nuclear envelope. They hold it together, they hold it in place, and they form cell-to-cell junctions. These are junctions that hold your skin cells tightly together to make sure that nothing is able to seep in. They also provide a protein called keratin that provides mechanical strength to skin cells. That's what makes them strong and a good barrier that prevents bad things from getting into your body. Microtubules. They are hollow cylinders made out of two globular proteins called alpha and beta tubulin. Spontaneous pairings of alpha and beta tubulin molecules form structures called dimers. The prefix di means two. Those dimers then arrange themselves into tubular spirals of 13 dimers around. The most important of these is a structure called the centrosome. These microtubules interact with motor molecules like kinesin and dynein to cause movement of organelles within the cell, and they form what's called the mitotic spindle, which distributes chromosomes during cell division. So whenever a cell decides, hey, I'm big enough, let's split into two, It copies its chromosomes, and then these mitotic spindles formed out of microtubules split the chromosomes up so that each new cell gets the correct number. So microtubule operation, so there is a vesicle, some kinesin, and our substance down here. The vesicle moves, not the microtubule. Here we have images of the cytoskeleton, okay, these long fibrous strands. We start at 850 times magnification here, okay. We move on to 1000 times magnification here with intermediate filaments, and down here with the microtubule, it's not listed, but it is very, very, very small. Centrioles. They are short, hollow cylinders composed of 27 microtubules that are arranged into 9 overlapping triplets. There is 1 pair of centrioles in each animal cell. And they're located in centrosome of animal cells only, and they are oriented at right angles to each other. Now this might sound strange, but that's important for the process of mitosis. And they help to determine the plane of division. They help figure out exactly where the middle of the cell is so that each new cell gets the right amount of organelles, the right amount of cytoplasm, so that each one gets what it needs. These centrioles may give rise to basal bodies of cilia and flagella. Basically, they provide an anchor point for those structures, which we're going to talk about in just a little bit. So here is the structure of the centriole. It has a hollow center, okay, and then here are those triplets. All right, here we have an actual image created using an electron microscope. All right, cilia and flagella. Cilia are hair-like projections from the cell's surface that aid in its movement. They are very different from prokaryotic flagella. They make up the outer covering of the plasma membrane, and inside this is a cylinder of 18 microtubules arranged into 9 pairs. In the center are 2 single microtubules, and this pattern, this template, is used by all cilia and flagella. In eukaryotes, the cilia are much shorter than the flagella. The cilia move in coordinated waves like oars. They line the lining of your respiratory system. They are how oxygen is exchanged inside your cells. And flagella move like a propeller or a corkscrew. And they are what propel sperm cells whenever they are on the move. So here is a diagram showing the structure of a flagellum, which is the singular form of flagella. So take a minute to study those. And that's all I have for you guys. If you have any questions, feel free to shoot me an email. I'll see you in class tomorrow.